So I have great pleasure in talking about the future of microbiome medicine and editor's perspective. I am the editor-in-chief of the journal Gut, and this is what you would see on the front cover of Gut each month. It's a selection of beautiful images that I select as a reward to the editor's choice. This is my favorite article each month. And as a reward to the authors, I select one beautiful image from their city. So I'm very proud to have been the editor of Gut for the last 16 years. And one thing, this privilege has given me the opportunity to indulge sometimes in my favorite part of the gastroenterology, which is this microbiome revolution. And I'm also very proud to have nurtured papers that perhaps not deal with gastrointestinal or liver disease, but outside of the GI tract, yet related to the gut microbiome. So I think these are kind of key papers that were published over the last 10 or 15 years or so. So we are all interested in the microbiome. This is a fascinating revolution in medicine and in science for the last 20 years or so. And rightly so, because we are essentially outnumbered by microbial cells. They are very metabolically active. They contribute to metabolism of the host, the immune system, and so forth, and essentially become a major interest of many scientists across the globe. Now, so this microbiome revolution that is occurring is a great opportunity to advance understanding of human health and disease, but we must do it right. And I think this is the kind of the take-home message from this particular lecture this morning. Now, this is perhaps the most important slide that I will put up this morning, because it shows you that the human body does not work in compartments. We tend to be too focused sometimes on our own system, our own kind of disease model and so forth, and ignore the fact that this is part of a collective in the human body. Each compartment in the human body has an impact on the others, and this interaction, this collaboration and being affected by the others is extremely important in understanding human disease. So I think this is a very important concept. What's happening in your gut will impact on your brain, will impact on your kidneys, the gut-kidney axis, the liver axis, and so forth. All of these things are interconnected and interrelated. Now, it was Hippocrates, in fact, that said that all disease begins in the gut, but in fact, it's the microbiology of the human body that is relevant to all of these processes. Now, how does this microbiome then exert such widespread effects? Well, the reason lies in the fact that each microbial cell, whether it's bacteria or fungi or archaea or so forth, is a little factory that is able to metabolize and ferment and basically handle all of the material that we give them in the gut, whether this is fibers, whether it's amino acid, xenobiotic compounds, and so forth. And they metabolize that and produce active metabolites. For example, the short-chain fatty acids or the activated post-drug modified xenobiotics and so forth. And these metabolites that are very important then act on host receptors and impact on host physiology and so forth. So this is really the essence of this microbiome revolution. We have little factories that metabolize the whatever we deliver to them and produce things that are metabolically active and impact on human physiology and pathophysiology. Now, the take-home message from this is that essentially we are all united in biochemistry, as my colleague, Professor Mark Morrison from Queensland coined this term. Now, we talked about these metabolites. The classic ones are things like short-chain fatty acids. These regulate epithelial barrier function. They have a profound effect on mucosal and systemic immunity. And although they are produced in the gut, they have direct and indirect effects on immunity at extra-intestinal sites, including, for example, the liver, the lungs, the reproductive tract, brain, and so forth, and have, of course, been implicated in such a variety of different human diseases. So the gut microbiome in health and disease, it's key to many aspects of human health, including immune, metabolic, cardiovascular, neoplastic, and neurobehavioral traits. And the most exciting part of this is that it's manipulable. We can actually change it. Unlike our genome with which we are stuck, we can actually do something to change our microbiome. And this has relevance to health and to disease and to treatment and to prevention of disease and so forth. Now, in this talk this morning, I would like to concentrate on a few aspects. I would like to discuss those key aspects that define the future of microbiome medicine. One most important one is the paucity or lack of longitudinal studies. and I'll talk a little bit about this. I will talk about the definition of the healthy microbiome, the role of diet and nutrition, the effect of medications that we take, and then finally talk about manipulation of the gut microbiome and some guidelines that have been published recently about the microbiome field. So let's start with these so-called longitudinal studies. Now, the holy grail in microbiome research, and this applies to all research related to health, is the ability to design a longitudinal study that starts, crucially, before the condition develops. So when there is a state of health, then the condition happens, then obviously there's an outcome to either the condition itself or its treatment. And ideally what you want to have is a baseline which starts before that condition develops. Now, during that baseline, you would like to collect all of the data, all of the kind of metadata, all of the biological markers, all the tissues, whatever is relevant to understanding that disease, and then wait for this condition to happen. And then, of course, study it during that condition, and then study the outcome after treatment or intervention. Now, as you can appreciate, this is almost impossible to do for conditions that take decades to develop, like cancer. For example, colorectal cancer might take 20, 30 years to develop. So clearly, it's not practical to have that design whereby you start at the precondition stage. There is, however, some examples that allow us at least proof of principle to apply that concept of longitudinal design. And one imminent condition is obviously pregnancy, because with pregnancy, you can certainly have a baseline preconception before the condition develops. You can collect all of your markers. You can collect all of the identifiers of future health and future outcomes of this pregnancy. You can monitor the pregnancy during its short period, which is 9 months, thankfully. And then, of course, you have the outcomes of that pregnancy, whether it's a completely healthy child and mom, or the complications that are well described, like preeclampsia, gestational diabetes, and so forth. So this was a great example to apply that longitudinal design. And we did this here at the Microbiome Research Center in Sydney by designing the Mother's Babies study. Now, this is a preconception study whereby we collect all of the markers, both genomic, microbiome, and all the other relevant physiological markers, as well as from mom and from the partner. Then we wait for the pregnancy to happen. So we have serial collections in the preconception stage. We take samples from trimester 1, 2, and 3, and then we follow the mums up to 2 years postpartum. And of course, we also look at the microbiome of the baby as it develops into a young child. Now, this is a beautiful design. It's a very difficult study and a very expensive study to do that. But it's one that would provide you with ideal opportunity to understand exactly what happens before these conditions occur, whether it's healthy outcomes or disease outcomes. So I think this is an exemplar of where we can apply this longitudinal design to understand outcomes of health and disease. So longitudinal studies are essential. Now think about other conditions where you can apply this one. I suppose if you were to pick another one, it would be infections. For example, we've just had the COVID pandemic, when it was affecting hundreds of thousands of people in each country. And you could have easily had a cohort of people that were unaffected and then follow them up. And sure enough, quite a large number of us all kind of developed this infection. So you could have identified the markers that would predict the outcomes of this infection, whether it's totally healthy and hardly any symptoms, to ones that ended up in ICU and had a mortality. So there are examples where you can understand the kind of using this longitudinal design. The second big issue in the microbiome field is the so-called definition of normal. What is a healthy microbiome? What is it that we aspire to have in our gut that could be associated with a healthy lifestyle and a healthy absence of disease and so forth? Now, believe it or not, this actually is one of the most difficult questions to answer. And there have been several attempts to define a healthy microbiome. We published this very nice review in Gut last year called What Defines a Healthy Gut Microbiome, and considered all the different kind of attempts at doing that one. You could start with the easiest one, the simplest one, if you're designing a study to say, well, a healthy microbiome is one where there is absence of disease. So I'm, for example, interested in studying inflammatory bowel disease. I would pick a control group that doesn't have inflammatory bowel disease. And this may be good enough for that particular question. However, you could also argue that those healthy people that you've included that don't have inflammatory bowel disease might still have some kind of illness that gives them, for example, bloating or irregular bowel movements or discomfort and so forth. So absence of diagnosed disease is no guarantee that you have a healthy, balanced gut health. And as such, this perhaps is not sufficient to have absence of disease as your marker for healthy microbiome. You could also move up to the next level and say absence of disease or digestive complaints. So these participants are healthy, they don't have any complaints of any GI symptoms and so forth. But again, we have no idea how healthy their gut barrier is, whether in fact their functionality of this gut microbiome is defined with optimal health and optimal outcomes. So these are the different kind of complexities that come into this question of defining a healthy microbiome. Now, again, at our Microbiome Research Center, we try to tackle this by coming to it from a different direction. So instead of looking at the microbiome and figuring out whether it has all the markers of a balanced, healthy, diverse microbiome, we thought, let's just ask the question, well, who are the healthiest people in our society? And perhaps if we can collect those, recruit these healthy people, optimally healthy people, we can then look at their microbiomes and see what characteristics or features unify them or define them. Now, to be able to do that, we looked and said, well, who are the healthiest people? And you could argue that if you look at, for example, elite athletes, well, they're incredibly healthy. They eat well. They exercise, obviously, to extreme levels. They're very strong, fit, lean. They have no disease. They're usually on no medications and so forth. And they would be very interesting to study. But again, you can argue that if you are an elite athlete and you're under stress of competition, that might impact on the microbiome. But it's still quite an interesting group to look at. But in parallel with that, we wanted to look at athletes or active, healthy people that do this for fun. So this is actually, it's their hobby to exercise regularly and to eat healthy and live a healthy lifestyle. And we wanted them also included in this search. You could still argue that both of these groups, even though they're healthy now, in kind of early adulthood, they might not be in 5 years' time. So which is the group of people that essentially has been tested by time? And then you come to the conclusion that it's these people that have reached advanced age, 95 plus, the centenarians who have survived all of the kind of pressures, both from their genetic background as well as the environment, yet retained quite a healthy phenotype, absence of dementia and so forth. So we decided to include them as well. And between all of these three groups, we were able to kind of define a core microbiome that unifies all of them. It's a unique kind of set of taxa that defines all of these different healthy phenotypes. I think this is a very interesting approach because it could essentially be the basis for your healthy optimal microbiome. And we use that as our template for manipulation. If you want to bring people back home, if you like, from a particular disease process, this is the kind of the template that we use for manipulation. So I think this is a great opportunity to define that. Obviously, it will have to be a multi-omic, multi-site, trans-kingdom definition. It would have to use artificial intelligence to be able to coordinate all of these multi-omic data. And this could then become an excellent way of formulating the basis for your intervention, including things like fecal microbiota transplants. So when you're dealing with the microbiome, of course, we're all familiar with the factors that impact on the composition and function of the microbiome, and these are very diverse. We're all very familiar with these, starting with the mode of birth, the feeding, whether it's breastfeeding or formula bottle feeding. Of course, the diet is the most important contributor, but equally, it's the geography, where you live, where you actually grow your food, the soil from which that food is produced and so forth is also relevant. And of course, medications, what we actually take routinely to either treat specific disease or for health supplements and so forth. All of these, of course, interact to impact on the composition and functionality of your gut microbiome. So take-home message from all of this is, again, this is a great opportunity. The microbiome is manipulable. You can change it quite easily by a variety of interventions. So let's start now with some of the kind of the recent literature on diet and the microbiome. I put this as number one because I think nutrition and diet is the most important aspect of this microbiome revolution. It's the most predictable, the most sustainable way of changing the microbiome. and for which we now have some amazing data that has been published. And over the last 12 months, we've had some key papers that I will highlight in the next few minutes or so. This was a paper that was published this year, Nature Microbiology, and looked at the kind of microbiome signatures in people that consume very different diets from each other, like vegans, for example, where it's all plant-based and there's no animal sources at all, like eggs or milk and so forth, compared to vegetarians who allow the animal kind of based products like eggs and milk and so forth, and dairy, and versus the omnivore diet, which is obviously very common across the globe. So they looked at this, and it was quite an impressive multinational metagenomic cohort with a size of more than 21,000 individuals. And then they were able to define those signatures that are associated with key components, big components like meat or dairy or fruits and vegetables and so forth. And not surprisingly, there was a difference. There was quite, dramatic difference between, the vegan signatures compared to the omnivore signatures and so forth. As you might expect, the vegetarians had a kind of a halfway between. But the crucial thing to also realize from this work is that those signatures were also impacted by what could potentially come from the food itself. So there's a microbiota that gets transferred through the dietary components that could form part of our own human microbiome. This is an interesting paper, and the take-home message from that particular study is that it reinforces how humans can shape their own gut microbiomes, and by extension, their health. That's directly through simple dietary choices. You can pick what diet you want, but also on a societal or a global level, by the way we produce the food. So agricultural practices that are used in this food production are also very relevant. So these are all important take-home messages from this particular study. Now, and this, of course, reminds us of the concept of One Health, because we don't exist in a vacuum. We live within this environment that has animals, it has plants, it has pollutants and so forth, and all of these things are obviously integrated into this One Health concept that is espoused by all of these World Health Organizations and so forth, trying to achieve perfect health and in unison with our environment. Another study that was quite a landmark study published last year in Cell, this looked at this interpersonal variations in the diet microbiome interactions. So we eat different diets, and they appear to have a different effect on our microbiome. And this wasn't really clear why until the study was published, and it was an excellent study that looked at how certain taxa, certain microbiome taxa, are able to, say, toxify non-toxic compounds, for example, by producing antibiotic or antimicrobial products. And this, of course, leads to microbiome disruption, whereas others can detoxify toxic compounds and lead to microbiome stability and resilience and so forth. And this really explains why the same dietary compounds can have different effects on the microbiomes of different individuals. So this is a very important concept. Yet another study also published this year in Cell, and this looked at the so-called restore diet. This is a non-industrialized type diet. So this is one that is devoid of all of the kind of the modern additives and supplements that we put into diet in the processing of diet, and going back to the simple natural products that we used to consume decades ago, but also combined it with one of the so-called lost microbes. These are perhaps ancient taxa that we no longer have in the human gut. Things like this L. reuteri, and that particular study showed how this L. reuteri persistence was associated with increased short-chain fatty acids. There was a microbiome diversity, decreased microbiome diversity. And essentially this restore diet, in addition to the lost microbes, was associated with a favorable metabolic outcomes. So this is the summary of the findings. And indeed that this diet redressed several microbiome features that were altered by industrialization. And this diet induced these beneficial changes to the microbiota-derived plasma metabolites, and it had a positive cardiometabolic health benefit. Now, this is very encouraging because, again, it just is pointing us towards optimal dietary practices and dietary compositions that should hopefully be developed across societies and so forth. Yet another kind of marker, another excellent study published in Nature Medicine, again this year. This comes from Africa, and this looked at the so-called African heritage diet versus Western diet. And what they took here is people who consume this African heritage diet. It's akin to the kind of non-industrialized, healthier one. It has more plant-based components and so forth compared to the typical Western diet, which is rich in meat and fats and sugars and so forth. And they did a crossover so that you start with, if you're on if you're a rural dweller on the African heritage diet, we take your baseline and you switch to the Western diet, and vice versa for the urban dwellers and so forth. But they also tested the effect of this fermented drink called mbege, which is essentially a beer made of fermented bananas and millet, and looked at the effect of these diets and the kind of crossover design, and showed very nicely that the switch from a heritage style to a Western style diet affected different metabolic pathways that were associated with non-communicable diseases. And in particular, it promoted a pro-inflammatory state. So this is what underpins most of these things like, heart disease or strokes and so forth, a pro-inflammatory state. So that switch from heritage to Western diet increased that propensity through the microbiome. In contrast, the switch from a Western style to a heritage style diet or consuming this fermented beverage, the mbege, had a largely anti-inflammatory effect. This is very nice kind of mechanistic data. Now, we move on from diet into something that is also incredibly important, and it's not, been kind of frontline news in the microbiome field until relatively recently, even though we should have really anticipated this decades ago. In fact, the assumption all along was that the drugs that have an impact on the microbiology of the human body are antibiotics. But this key paper, and I think this is one of my favorite papers of all time, this was published in 2018 by Meyer et al. In Nature, and this looked at the extensive impact of non-antibiotic medications on the human gut bacteria. So they tested more than 1,000 non-antibiotic medications on a panel of 40 commensals, and showed, in fact, that 24%, a quarter of all these non-antibiotic medications, has a direct effect on these commensal microbiota. And amongst the groups of drugs that had the maximal effects were antipsychotics. And of course, we're very familiar with the side effects of antipsychotics. You develop metabolic problems, obesity, diabetes, heart disease, strokes, and so forth. And this is the most dangerous aspect of the condition. Second was the proton pump inhibitors. These are the powerful acid inhibitors that are incredibly widely prescribed. Almost 10 to 15% of humans are now on long-term PPIs, which is quite a staggering statistic. And of course, what the PPIs do is that they take out your gastric acid, and that is really your first line of defense against ingested microbes. So it makes sense that these PPIs have a direct effect on manipulating and changing the gut microbiome. So this is emerging as a very, very key manipulator that has negative outcomes. And finally, the painkillers like morphine and so forth. And we're all very familiar with the opioid pandemic and how it's, this is a vicious cycle. It turns out to also have a very profound effect on the commensal healthy microbiome. So this is a kind of a wake-up call about the effects of medications. Now, it works both ways because there are drugs that have a beneficial effect. For example, metformin. This is a medication that we use for type 2 diabetes. It's commonly used also for other conditions like polycystic ovary syndrome and so forth. Essentially a metabolic kind of disease that you're dealing with. And it has a very, very clearly defined effect on the microbiome. This is a beautiful paper from 2018 showing how it works and the mechanism by which it benefits the host. And this other paper that was published earlier this year, perhaps less known, and in a less prominent journal, but I think it's just as important because it showed for the first time a potential mechanism by which metformin can actually lead to a change in this gut microbiome. And this is because the metformin-regulated glucose flux allows the glucose to come from the circulation into the intestinal lumen, where it interacts. There's a symbiosis with the gut microbiota, and this perhaps mediates the positive effect, the beneficial effect of metformin in human disease. This is an editorial that was published late last year talking about what everybody's talking about now, which is the kind of the obesity drugs, the GLP-1 receptor agonists and so forth, and how they're emerging as being beneficial to so many different things. Why do these obesity drugs seem to treat so many other ailments? Now, no one has looked at this yet, but I think there are a number of groups looking at it and asking about the effect of GLP-1 receptor agonists and equivalent drugs on the gut microbiome. There is every reason to suspect that it's relevant because there's physiological benefits of the gut peptides and the gut microbiota metabolites. This well described. This is kind of the normal physiology. It's also clear that the gut microbiota metabolites promote GLP-1 production. So there's a very intricate kind of bidirectional connection. So far as I know, there's only been one animal study that has looked at the effect of some drugs like semaglutide, for example, in a diabetic mouse model, and showed that in fact the semaglutide improved the gut microbiome in these diabetic mice. And this improvement of the gut microbiota may be an important reason for the treatment and prevention of NAFLD with semaglutide. So this is very promising, and I think it's early days, but it would be fascinating to see how this kind of field pans out. So the take-home message is that medications have a direct effect on the microbiome. These effects may explain the positive as well as the negative effects of drug treatment. Now, why is this relevant? It's because in the future, and this is the future of the microbiome medicine, we may be able to actually predict who is going to respond to what treatment, what side effects might you get if you go on a particular treatment based on your biomarkers related to the microbiome. So I think this is something that is very, very exciting. Now, other very fascinating papers have been published also in the last year or so. This is the paper published in Cell showing, in the context of the Framingham Heart Study, a very famous study that looked at cardiovascular disease, and showed that certain taxa like Oxalobacter species that are associated with decreased blood and stool cholesterol. It's almost like, you've got these microbiota that act as your natural statins because they're equipped with essentially the machinery, the genome that allows it to metabolize cholesterol. Now, this is fascinating and may at some point, become a next generation probiotic that could be a supplement to reduce your cholesterol. You never know. I think this is something that is definitely worth trying. Equally, this other very, very interesting and amusing paper that was published in Nature in 2022 showing that certain microbiota protect against the effects of smoking. So these, like Bacteroides xylani solvents, for example, in a NASH or fatty, non-alcoholic fatty liver disease model, actually metabolizes nicotine. So it kind of degrades the nicotine and protects the mouse from developing the full phenotype of this fatty diet and liver disease and so forth. Finally, I think another very, very beautiful example is aspirin, because aspirin is very commonly prescribed. It's either given as a chemopreventive agent, so you take it, it decreases the risk of developing cancer, particularly colorectal cancer, but it's also used for cardiovascular risk protection. Yet it only works in 50% of cases. So the maximum effect that it might do on a population level is 50%, and it's never been clear why. We published this paper. This is Professor Jun Yu's team from the Chinese University of Hong Kong. A beautiful paper. This was an animal model of colorectal neoplasia and showed that, in fact, it was a dual effect. So aspirin leads to enrichment of beneficial bacteria that mitigate the development of colorectal neoplasia in those murine models of colorectal cancer. But equally, there were some taxa, like the Lysine sphericus, which actually degrades aspirin and makes it less bioavailable, so that it dissipates the positive effect, the positive chemopreventive effect of aspirin. So it could be both ways. It could be that some people have bad microbiota that dissipate the effect of aspirin, while aspirin itself leads to the proliferation of healthy ones that can protect you. Now, it'll be interesting if this actually pans out in humans, and I think there are studies that will show this very soon. So let's look to the future now. How can we manipulate the gut microbiome? I mentioned at the beginning that the beauty of this field is that we can change it. We can change the microbiome and impact on human health. So are there ways of doing it? Well, yes, there are. There are many, many ways of doing it. I mentioned diet. That's the most important and the most durable. Exercise does that as well. So it changes your microbiome to a healthy one. You can use a variety of prebiotics, probiotics, symbiotics that deliver certain types of benefit to human health. You can use antibiotics to get rid of certain bad bacteria and so forth. Or you can use genetically modified microbiota that deliver specific products. And I think this is a fast developing field as well. And finally, you can change the whole lot by giving, this is the fastest way of changing your microbiome, by transplanting somebody else's microbiome into your gut. This is fecal microbiota transplantation. So diet, I've mentioned, I talked about very, very extensively. And the concept of food as medicine, I think, is gaining momentum. And I think this is something that will become much more kind of practiced in the near future because of its durability and its impact and fantastic evidence base to show its benefits. All of the biotics that have been developed are now well established, and there is a lot more science that goes into the verification of certain what we call next generation probiotics, because these are the ones that have been validated and their mechanism is understood, and they will have undergone or undergoing clinical trials to show to show their benefit as modulators of human health. We've also got the so-called live biotherapeutic products, and all of these things are coming now and with very good science. This is an example of a next generation probiotic championed by our colleague, Professor Patrice Cani in Belgium, the Akkermansia mucinifera. This is a paradigm for next generation beneficial microorganisms. There's very good evidence. This is an excellent review published last year showing the effects of Akkermansia mucinifera. And you will see there that it has these so-called Dr. Jekyll and Mr. Hyde. So it has beneficial effects in so many conditions, but for others it might be bad because it's skewing the immune response in a different direction and so forth. And this is something worth remembering, that there are kind of dual functions, and depends on the context of how to use these probiotics and how their effectiveness might be. Now, the other fascinating kind of development, certainly over the last 2 or 3 years, is been the use of bacteria or microorganisms to deliver products, to deliver things that are useful in certain contexts. Now, this is an example of delivering tumor-specific neoantigen-derived epitopes. And this is in the context of increasing systemic anti-tumor immunity. So this is absolutely fascinating work that is developing very fast. And more examples. This is just an editorial on that paper showing the value of using Escherichia coli in Nestle 1917. This is a famous probiotic. And this is used as engineered, so it becomes a delivery of neoantigen for cancer immunotherapy. And this was tested in these murine models of colorectal cancer, as well as malignant melanoma. So there's a lot of promise in these kind of approaches whereby you deliver those engineered microbiota to specific intratumor environment and so forth. This is another one. This is Lactobacillus plantarum. This was published this year in Cell. And this was actually administered intranasally. And so it's an intranasal administration. It's engineered to produce leptin, for example. This is a hormone that, obesity regulating kind of leptin. And it basically gets taken up through the kind of deep nasal epithelium. And this is fascinating in the context that it is possible to deliver things, close to the brain and gets picked up by the brain. But it also opens up the possibility of understanding why certain things that we have never understood that are associated with things like dementia. I mean, there's always been an association between, the oral microbiome and dementia, or the kind of sinusitis or middle ear infections and these kind of brain-related conditions. That may be a similar mechanism. So I think it's just pointing us in the right direction to try and understand pathogenesis of disease, as well as potentially offering a therapeutic approach to delivering products that could be taken up by the brain. Now, yet another one. This is another excellent example. This was also published this year. This is now using Cutaneobacterium acnes. This is the old Propionibacterium acnes, and this is to kind of deliver something that would give you ultraviolet stress, oxidative stress protection. So it's an antioxidant that is secreted by this C. acnes, and it reduces that oxidative stress in this UV stress model. Fascinating work. Now, finally, FMT, and I think FMT is going to give us lots of clues about how to treat disease, but it's a very kind of blunt approach. Essentially, it's random at the moment, and that's because we don't have a proper definition of a healthy microbiome. Going back to what I said at the beginning. But if you use an intelligent approach where you're actually matching the disease with the potential donor, I think that might be something that is very, very exciting in the future. And this is something where a lot of work is being done. Now, finally, in the last just 2 or 3 minutes, I would like to alert you to some key guidelines that have been published. This is just for general reading. Very important because it kind of defines the rules of the game, if you like. This is the one that was published this year, and this was a consensus statement on establishing causality, therapeutic applications, and the use of preclinical models in microbiome research. It's a very, very worthy effort. So this was published in Nature Reviews Gastroenterology and Hepatology, and it's an excellent review. And it's it's a consensus statement, and I think it's worth reading and worth incorporating in your study designs and so forth. Next was this excellent review also in Nature Medicine on clinical translation of the microbiome research, again written by some of the luminaries in the microbiome field. This was an excellent paper that we published in Gut dealing with the development of live biotherapeutic products, and this was a position statement from the Asia Pacific Microbiota Consortium. And this very interesting consensus paper that was published in Lancet Gastroenterology and Hepatology. This was an international consensus on microbiome testing in clinical practice, because you'll no doubt be familiar of the proliferation of straight-to-consumer microbiome testing. This is commercially available, and this consensus kind of statement lays the rules of how we handle this and how we deal with it, because I think this is a very important concept. And you'll see the description there. And finally, this just published literally last week. This is the microbiome for clinicians. And again, an excellent review that gives you kind of state-of-the-art where we are essentially in terms of incorporating microbiome into clinical practice. Now, this is something that I'd like to highlight, because this really is the crossroads for the microbiome field. And I think we're at the stage when we should be thinking very seriously about incorporating into clinical practice, but we should do this in a very controlled way. Now, there is no doubt that this will be heavily dependent on artificial intelligence to be able to handle the magnitude of data that is generated by microbiome testing, by genomic testing, by all of these metadata and so forth. This cannot be done. It has to be done through very, very well-designed artificial intelligence pipelines and tools and so forth. Now, but it's a great opportunity because it allows us to understand mechanism of disease, but also how to offer treatment. And I think we're not quite there yet, because we still have these number of key things to develop. A proper definition of a healthy microbiome, some longitudinal studies that allow us to at least understand the proof of principle of how things are before the condition develops. And of course, ultimately, we have to all collaborate to do this, which is a state-of-the-art, multi-omic, multi-site, trans-kingdom, validated mechanistic studies. Now, this sounds too prohibitive, but it's not. This is something that is achievable if we can pool our resources and work together and try and solve something once and for all, instead of doing an incremental approach to science and to medical research. So the future of microbiome medicine is bright. I think the buzzword is going to be personalization. Personalization in every aspect of that health journey, whether it's diagnosis, treatment, prediction of side effects, prognosis. There'll be personalized nutrition, there'll be personalized medicine and medications and therapeutics, and we have a very wide array of things that we can use for this, and I think this is really, really very exciting. And my final take-home message is that this is a new frontier. It's very exciting. We've got to prepare for it very carefully. We've got to pool our resources and collaborate, and we've got to put pressure on our politicians to actually invest in this field, because I think the return will be massive, and it'll be more than anything that we've ever experienced. So thank you very much for your attention.